Hi Dr. Choi, this is our molecular biology primer project done by YueMin, Caroline Au, and Chloe Ooi. So this is our content. I'll be doing the introduction, YueMin will be doing materials and methods, and Chloe will end with the results and discussion. So this is a schematic diagram showing how Staphylococcus aureus acquires resistance to methicillin and its ability to express certain virulence factors such as surface protein adhesins, cytolytic toxin, and enzymes because of the activation of certain chromosomal genes. So the MAC-A gene in particular is located on the SCC-MAC and encodes for abnormal beta-lactam insensitive penicillin binding protein, PBP2A, which continues to synthesize new cell wall peptidoglycan even when the normal penicillin binding proteins are inhibited. So this is our Staphylococcus aureus strain MS4 complete genome DNA template, which we'll be using to design the primers. But before that, the purpose of amplifying the MAC-A gene for PBP2A is because we want to detect the MAC-A gene for PBP2A in human DNA so that we are able to identify methicillin-resistant Staphylococcus aureus, MRSA. And from that information, the doctor is able to prescribe alternative antibiotic to methicillin and certain beta-lactams to the patient. So these are some primer guidelines. It is wise to avoid GC3'N because having 3 or more G or C bases at this position may stabilize non-specific annealing to the primer. Next is the primer length. Usually a primer length of 18 to 22 bases is optimal for most PCR applications because shorter primers could lead to the amplification of non-specific PCR products. Thirdly, melting temperature. Usually good results are obtained when the melting temperatures for both primers are similar within 2 to 4 degrees Celsius and in the range of 55 to 65 degrees Celsius. Fourth point, GC content of the primer should be between 40 to 60%. And the last point is to avoid sequences that is likely to form internal secondary structures, such as intra-primer homology, which means that more than 3 bases that complement within the primer, or inter-primer homology, which is the forward and reverse primers having complementary sequences, as these circumstances can lead to self-dimers or hairpins or primer dimers instead of annealing to the desired DNA sequences.
Now I'll be going through the materials and methods that we have used to create the primer. First, we used NCBI to search for the gene sequence of PBP2 and the mutated PBP2, which is the PBP2A gene. Upon acquiring the gene sequences, we used this website EBI to locate the point mutations via pairwise alignment. Next, we used GeneRunner to locate the exact site of the point mutations. The numbers below the start is the exact location of the point mutation. Lastly, we used Primer Blast to create possible primer pairs for our PCR. We also set the melting temperature range according to our guidelines.
So now I'll be talking about the results. So the analysis of the forward primer that we found only gave us 0 hairpin loops and with only 1 dimer. And the analysis of the reverse primer gave us the results of 2 hairpin loops and 5 dimers. And with all the hairpin loops and dimers melting temperatures accounted for to be lower than the primers, we can safely conclude that our primers are suitably apt to be used in PCR. In the end, we were able to amplify with our design primers a PCR product with a sequence and length of 598 base pairs, within which included 2 distinctive point mutations characteristic of the gene sequence used to code for the penicillin binding protein 2A that gives MRSA its methicillin resistance. Thus, we were able to discern and identify from PCR the particularly methicillin-resistant strain of S. aureus that causes so much harm. As for our discussions, the difficulties that we faced included finding the exact sequence of MAC-A gene that codes for the original PBP2 protein and the mutated PBP2A protein. Furthermore, we faced the challenge of identifying the location of the point mutations in the PBP2A gene sequence that were distinct from the normal PBP2 gene sequence. Other than that, we also had the challenge of selecting which primer to use. And so how do we select which primer to use? The first feature that we considered was that the PCR product cut must include the point mutation. Second, that it cuts the shortest PCR product length possible. In other words, it has to cut the shortest PCR product length that includes the point mutation.